Good morning students, welcome back to our biology class. We were discussing in our earlier classes sexual reproduction in flowering plants, that is chapter 2 of our syllabus, and today we are going to discuss about the embryo. Okay, embryo is the post fertilization event which happens after fertilization. So in our last class we already discussed what are the three structures that are formed after fertilization. First is your endosperm, second is your embryo, and third is your seed. Okay, so this we were already discussing in our last class. Today we will discuss what is embryo. Embryo develops at the micropylar end of the embryo sac where the zygote is located. Okay, so while discussing your megasporogenesis, we discussed the structure of ovary. You remember students? So this is the structure of ovary. This was the micropylar end, this is the callosal end, and in micropylar end only here you had this egg cell and these two were the polar nuclei. Okay, so you already know that one male gamete, what it has done, one male gamete, it has come and it has fertilized this egg cell. Okay, and another male gamete, one more male gamete, it has come and it has fertilized these two polar nuclei. Okay, so these two polar nuclei plus this male gamete, they has formed trisome and this is endosperm. Okay, and this male gamete and this egg, when they will be meeting, they are going to form this zygote. Okay, and the zygote later on it will form a embryo. So where it is from where it is being formed, the embryo, it is always being formed from the micropylar end. Micropylar means this end is called micropylar end, this lower end. Okay, lower end of the embryo, it is called micropylar end, and always the embryo construction or formation of embryo, it is always happening in the micropylar end. This is a very important thing students, you need to note down. The embryo, it is always formed in the micropylar end of the ovary. Okay, so this is the diagram of a structure of an ovary. Okay, so in your micropylar end, that means that lower end of the embryo sac, in that portion only your embryo is developed, where your zygote is being located. How the zygote is formed? After the fusion of one male gamete with your egg. Okay, and embryogeny is the early stages of embryo development. The zygote gives rise to a pro-embryo and subsequently to a globular, heart-shaped mature embryo. Okay, so what is the meaning of embryogeny? Embryogeny are nothing but it is the early stages how an embryo gets developed. Okay, the early stages of embryo development is called embryogeny. And what happens? First, the zygote, which is being formed after meeting of one male gamete and the egg. When one male gamete, that is pollen, when this pollen, it will fertilize this egg. Okay, your zygote is formed. Understood? So this zygote, what is going to happen to it? The zygote, it is give rise to a pro-embryo first. Okay, this zygote, it will give rise to a pro-embryo. It is not mature. Okay, it is going to give rise to a pro-embryo, and when this pro-embryo becomes mature, it becomes a globular, heart-shaped mature embryo. Okay, the embryo becomes heart-shaped when it becomes mature, and when it is not mature, it is called your pro-embryo, and it becomes globular as well when it gets mature. Okay, so these stages of embryo development is called embryogeny. Okay, now let us discuss what are the parts of an embryo. The parts of an embryo are embryonal axis, cotyledons, plumule, and radicle. Okay, these four are the most important parts of an embryo. The first one is your embryonal axis, second one is cotyledons, third is your plumule, and fourth is your radicle. Okay, so this from here you can see it is the structure of an embryo, and here this portion it is called plumule, and these two are the cotyledons. Okay, this is your radicle and this is root cap. Okay students, so let us understand what are different parts in detail. Okay, so this is from your textbook, all of you just pay attention to this. Embryo development always occurs in the micropylar end, as we have already discussed, of the embryo sac where the zygote is situated. Most zygote divide only after certain amount of endosperm is formed. Okay, zygotes are not going to divide very easily. They will divide only when the little amount of endosperm is formed. After that only it is going to divide. The reason is that this endosperm, it is going to create as a food reserve. If the zygote develops before the endosperm develops, this zygote will have to starve, okay, without food. So this endosperm, it is going to provide food. This is an adaptation to provide assured nutrition to the developing embryo. If the embryo wants nutrition, first the endosperm will have to be developed. That's why in our last class we discussed endosperm before discussing embryo. Is it clear, students? Though the seeds differ greatly, the early stages of embryo development are similar in both monocotyledons and dicotyledons. Okay, embryogeny, that means the early stages how embryo develops, it is generally same in case of both monocotyledons and dicotyledons. Okay, but the seeds may look different, but they are same. All of you, do you know what is the meaning of monocots and dicots? Monocots are nothing which have one covering over their seed. Okay, suppose this is a seed, if it has one covering, it is called monocot. If it has a double cover, it is called dicot. Okay, monocot means only one coat is there, and dicot has double number of coats, double layer of coats. So their seeds in monocots and dicots both are different, but the way how it develops, the early stages, that is your proembryonic stage, these are same. The zygote gives rise to a proembryo, as we have already discussed, and it forms later on, it becomes a globular, heart-shaped, mature embryo. Okay, a typical dicotylaninous embryo, dicotylaninous means having two cotyledons, okay, consists of an embryo axis and two cotyledons. See this diagram if you see. Okay, so this is a dicot embryo. So here you can see two cotyledons are there. Can you see two cotyledons, two covering? Okay, these two are two cotyledons. If it was a monocot, it would have had only one cotyledon. Okay, since it's a dicot, so it is having two cotyledons. And it has an embryonal axis. Embryonal axis is that portion of the embryo surrounding which the embryo develops. Okay, suppose this is the embryonal axis. This embryo is going to develop by taking the help of this axis only. It's just like the axis of a globe. Okay, as soon as the embryo will become bigger, it is going to take this as axis. Okay, it is going to take this as the center, then it is going to develop. The portion of the embryonal axis above the level of cotyledons is your epicotyle. The portion of the embryonal axis just above those cotyledons is your epicotyle. Above the embryonal axis, you are going to have your epicotyle, and hypocotyle, it will be below the embryonal axis. Okay, students? Just you see here, which terminates with the plumule or stem tip. The cylindrical portion below the level of cotyledons, it is your hypocotyle. The terminates at its lower end at the radicle or root tip. The root tip is covered by root cap. Okay, so this, as we know, these are the parts of your embryo. First is your embryonal axis, second is your cotyledon, third is your plumule, and fourth is your radicle. Okay, if you see this diagram, these two are your cotyledons. Your embryonal axis, it will be in the middle. Okay, this is your embryonal axis, and this upper portion of the embryo, it is your epicotyle. Okay, students, and this hypocotyle, it is the lower portion, and it is going to stick always to the radicle or root stem. Okay, and this is always covered by your root cap. Is it clear? So that is the thing this paragraph is trying to tell you. Embryos of monocotyledons, they possess only one cotyledon. Okay, only one cotyledon is there in embryos of monocots, and in the grass family are the monocots, and there you can see only one cotyledon is present, and that is called your scutellum. That is situated in the one side of the lateral of the embryonal axis. So this second diagram, it is going to show you the structure of a monocot embryo. Okay, so this is your monocot embryo. It has only one cotyledon. You see here, two cotyledons were there, two coverings were there. Okay, this between this white color portion, this is your embryonal axis. Here what is happening, only one coat is there. Okay, one coat is there, one cotyledon is there, that is called scutellum. Is it clear? And this root cap, radicle, this portion, this will always be there, like other the dicot embryo also. Okay, so generally in embryo you are going to see these parts only: embryonal axis, cotyledon, plumule, and radicle. These are common in both of them. Okay, plumal and radical, these are going to be present in both of them, but what is the difference is that in monocot embryo, see, this is LS of embryo of grass, this is monocot. Okay, this embryo is monocot and this is your dicot embryo. In monocot embryo you are going to see only one cotyledon present, and that is called your scutellum. Okay, and here you are going to see two cotyledons present. This is the major difference between the both monocot and dicot embryo. Okay, since we are talking about your monocot embryo, at the lower end of the monocot embryo, what is going to happen? You are going to have an embryo axis, and in that embryo axis, the radicle and the root cap which is present, it will be always enclosed or covered in an undifferentiated sheath. In one particular covering, both of them, this radicle and this root cap, they will be together, they are going to be covered in one particular sheath, and that is called your coleohyza. Okay, so here if you see in this diagram, you can see which portion is called coleohyza. You see this is a monocot embryo, radicle and root cap, they are together held together in the coleohyza. Okay, so the coleohyza, it is an undifferentiated sheath which is going to hold together this radicle and root cap. Okay, since all of you know that epicotyle, it is an important part of the embryo. Cotyledons you have already known, plumule you have seen, radicle you have seen, embryonal axis also you have seen. This is the axis in case of dicot, and in case of monocot this is the axis. Okay, here what is happening in the monocot, just above this scutellum. Okay, the scutellum, what is the meaning of this scutellum? This is nothing but it is the cotyledon. Okay, just above this scutellum you are going to have your epicotyle. Epicotyle, it will be present here itself, just above this scutellum, and below this scutellum you are going to have two hollow-like structures which is called coleoptile. Two hollow-like structures. Okay, these two are hollow-like structures. Hollow-like structures, these are called coleoptile. Okay, so we are going to read what this textbook is going to try to tell us about coleoptile. The portion of embryonal axis just above the attachment of the scutellum is called epicotyle. Just above the attachment of the scutellum, just above the attachment, the embryonal axis just above the attachment of scutellum is called your epicotyle. Okay, students, and you see here epicotyle has a shoot apex and a few leaf primordia enclosed in a hollow foliar structure called coleoptile. So coleoptile is a hollow structure in which what is happening, the shoot apex of the epicotyle, it will be present. Along with the shoot apex, who is going to be present? Few leaf primordia is also going to be present in this coleoptile. This coleoptile, it will hold the shoot apex of your epicotyle, and it is also going to hold the leaf primordia. Okay, so this is an important point, you can note it down. It is going to hold your shoot apex. It is going to hold your shoot apex and also few leaf primordia. It is going to be held in a hollow foliar structure which is called your coleoptile. Okay, so this is just an experiment. What to do? You just soak few seeds of suppose for example pea. Okay, this is very easily available as we don't have wheat and maize seeds here, very easily available. Or you can also take groundnut and you can keep it overnight. When you keep this groundnut or these things overnight, next night what do you see? In next night what you are going to see is that, suppose this is a seed, I have kept it, suppose this is a pea seed, I have kept it in water for whole night. After whole night you will see that this covering of this seed pea is going to come out. That is nothing but that is your cotyledon. Okay, that is your cotyledon which is going to come out. Even if you soak groundnut also, it is going to come out. Okay, so since these are dicots, so you will see two number of cotyledons coming out from that